Hello, my name is Naila Rahman,
And my name is Jordan Ramnarine.
and we are co-first authors of this article recently published in the Journal of Magnetic Resonance Imaging. Here, we will provide a brief overview about test-retest reproducibility in vivo, magnetization transfer ratio, and saturation index in mice at 9.4 Tesla. For more details and complete results, please refer to our paper in the JMRI website. Before we begin, I'd like to acknowledge our sources of funding, which include the New Frontiers in Research Fund, the Canada First Research Excellence Fund, and NSERC and OGS graduate scholarships.
Magnetization transfer, or MT, imaging has been used extensively to investigate changes in myelin content and integrity in brain development, injury, and white matter diseases, most notably in multiple sclerosis patients. MT is a contrast mechanism in tissue that is based on cross-relaxation or chemical exchange between protons in bulk water and rotationally immobilized macromolecules. MT contrast is invoked by application of an off-resonance radiofrequency prepulse, or MT pulse, to selectively saturate the spectrally broad macromolecular proton pool. MT is then observed as a reduction in image intensity.
Its normalized value, the MT ratio, MTR, is commonly taken as a measure for the strength of the MT effect and interpreted as a surrogate parameter for macromolecular content or myelination. MTR can be acquired with two scans, a proton density weighted or PD weighted scan with MT pulse off and an MT weighted scan with the MT pulse on.
Although MTR has been shown to correlate well with histological myelin content, it is also sensitive to the choice of sequence parameters, flip angle inhomogeneities, and longitudinal relaxation time, T1, mitigating the power of MTR as a measure of myelin.
Magnetization transfer saturation, MTsat imaging, was developed to improve MTR by decoupling MTR from T1 effects while maintaining a feasible scan time, and the theory is explained in detail by Helms et al. In MRM. MTsat requires an additional T1 weighted scan.
MTsat, being more independent of system parameters in T1 weighting and less susceptible to inhomogeneities of the receiver coil and the transmitted radio frequency field, provides greater specificity and contrast compared to MTR. For example, Lima et al. Reported that MTsat correlated with more disability metrics than MTR in patients with multiple sclerosis. Although previous in vivo MTsat studies have shown a high reproducibility in humans at 3 Tesla, the comparability of MTR and MTsat reproducibility has not been fully evaluated. This leaves the question of MTsat reproducibility in a preclinical setting at an ultra-high field strength.
The purpose of this work was to assess test-retest reproducibility of in vivo MTR and MTsat in mice at 9.4 Tesla and provide estimates of required sample sizes, which is essential in planning preclinical neuroimaging studies involving models of disease and injury.
All animal procedures were approved by the University of Western Ontario Animal Care Committee and were consistent with guidelines established by the Canadian Council on Animal Care. 12 adult C57 black 6 mice, 6 males and 6 females, were scanned twice, 5 days apart. Before scanning, anesthesia was induced with 4% isoflurane. Following induction, isoflurane was maintained during the imaging session at 1.8%. The mouse head was fixed in place using ear bars and a bite bar to prevent head motion.
Imaging was conducted on a 9.4T Brooker system, and these are all the scan parameters which were used to acquire the T2-weighted TurboRARE anatomical imaging and the MT imaging with Flash 3D sequence. A B1 map was also acquired to correct for local variations in the flip angle. So MTR and MTsat maps were generated using in-house MATLAB code, and the code and dataset are available online. MTsat maps were calculated following the original method proposed by Helms et al. And outlined by Hagiwara et al. Image registration was performed using affine and symmetric diffeomorphic transforms with ANS software. The region of interest masks were acquired from the labeled Allen Mouse Brain Atlas. For ROI-based analysis, the MT-weighted images were registered to anatomical T2-weighted images, and the T2-weighted images were registered to the atlas. And ROI masks were brought back from atlas space to the MT-weighted space by using the inverse transforms from these registration steps. And then the masks were manually edited as needed. For voxel-wise analysis, the MT-weighted images were registered to one chosen MT-weighted image.
We employed multiple statistical tests to explore the data. First, myelin plots were used to show the distribution of mean values. Next, a Bland-Altman or BA analysis was performed for the ROI-based analyses to identify any biases between test and retest measurements. For both analysis techniques, the scan-rescan reproducibility was characterized by the coefficient of variation, or CV. Sample size calculations were performed based on CVs from the ROI analysis.
Here are parameter maps that were used. So there's anatomical T2 weighted image, a magnetization transfer weighted image, a reference T1 and proton density weighted image, as well as a B1 map acquired from the scans. And here we show the ROIs overlaid on a MT weighted image. So you can see the cortex, corpus callosum, hippocampus, the thalamus, and the internal capsule. And these are MTR and MTsat images. So MTsat revealed slightly greater contrast than MTR between gray matter and white matter, which was noticeable when comparing the corpus callosum and the internal capsule, which are both white matter regions, to the surrounding gray matter. The normalized contrast between gray matter and white matter regions averaged over all subjects in MTsat was significantly higher than in MTR.
Myelin plots depict the distribution of the mean values for each metric within the ROI. BA plots revealed negligible biases with mean biases of 0.009 and 0 for MTR and MTsat respectively. Overall, MTsat exhibits slightly higher CVs than MTR, which may arise from noise propagation through the equations used to calculate MTsat. In general, CVs were comparable across all ROIs, as no significant differences were found between ROI-based mean CVs.
The voxel-wise CV maps showed very high CVs in the cerebrospinal fluid, or CSF, due to the low values of MTR and MTsat in the CSF. Throughout the whole brain, between and within subject CVs showed good reproducibility, with CVs less than 20%. And as observed in the ROI-based CVs, the MTR exhibited significantly lower between subject and within subject CVs compared to MTsat.
Sample sizes required are shown for each ROI given a different detectable effect. Both MTR and MTsat can detect small changes, less than 10%, with feasible preclinical sample sizes.
So in conclusion, we demonstrated that MTR and MTsat were reproducible in both ROI-based and voxel-wise analysis. And importantly, MTsat exhibited comparable reproducibility to MTR and could detect small changes with sample sizes of 15 to 20, while providing better contrast and maintaining a feasible scan time.
Thank you for listening, and we hope you check out our full paper on the JMRI site.